Welcome, everybody. I'd like to have you help me welcome Dr. Erica Forsberg. She has joined Bruker Scientific in 2020 to spearhead the growth of metabolomics in the Americas after an academic career at San Diego State University, where she focused it on untargeted and targeted methods of bioinformatics and of the gut microbe. She was a postdoc researcher in the Gary Shoestack's group at the Scripps Research Institute, where she was expertly trained in untargeted metabolomics and in multi-omics data integration. Please join me in welcoming Dr. Erica Forsberg.
Thank you. I feel like I'm missing half of my audience over here, so I might try and be creative here. What's going to be best? Nope, I think this side will be better. So thank you, everybody, for coming today. Yeah. I can, because I'm short. Last person must have been tall. Oh no, this is good. I think I can now see everybody, which is what I would prefer, to have a little bit more interaction with our audience here. So as I didn't even catch your name. Mason kindly introduced. I am the Vice President of Metabolomics at Bruker Life Sciences Mass Spectrometry. We make lots of really cool analytical instrumentation, and if you want to learn more about what we have, your local sales representative Raj is sitting up there, and our regional sales manager Corey Lytle is also here. So thank you guys for driving me around today and bringing me here to Texas A&M. So I will start by kind of giving you an introduction to our mass spectrometry portfolio for doing small molecule research. I actually use this little QTOF up at the top left called the Impact BIP, even in my own research and at the Scripps Research Institute, doing a plethora of untargeted metabolomics that even included some crazy nutritional experiments that I was running on organic versus non-organic fruits. So that was actually a pretty cool little small experiment that we were doing just to do sample prep kind of work. And then Bruker kind of found me in the middle of the pandemic and told me about this wonderful new instrument called the TIMS TOF and how it can really change and expand what you can discover in an untargeted metabolomics experiment. So in 2021, I did jump ship from academia and moved to industry, to the dark side, and became an expert at trapped ion mobility, which is what most of these instruments, except the big one in the middle, that's an FTICR instrument. But most of the instruments that I tend to work with these days involve some kind of ion mobility. And I will go into how that works in our systems in a little bit. But we also have a very comprehensive suite of software that goes along with that in order to easily analyze your data. So we like to make sure that when you get an untargeted metabolomics experiment, there's tons and tons of information in there. How do you actually characterize all those things? That's what the software team does. And they make sure that life is easy for you. So I will go over a little bit about metabolomics, and of course, within the central dogma of biology, we have our genes that translate into our proteins, and that goes into our metabolites. And we really care about the metabolites because they are closest to our expressing phenotypes. And whether that's in a molecular biology experiment where we're looking at human studies, mouse models, and cell cultures, or plants, whether we're looking at food and nutrition, or how medicines are metabolized, or even environmental exposures and how that gets incorporated into the metabolome, we end up wanting to get a picture of that metabolome. And we can do that through a number of analytical tools, including things like NMR and mass spectrometry. And that overall goal is to get what we call a metabolic fingerprint or profile. And this is what gives us some kind of indication as to what is in our sample on the metabolic level, as well as how things are changing between different sample cohorts. So in my title, I do mention that we are looking at a complex metabolome, and today I will be focusing a bit more on plants. And we've done some developmental work within our applications development lab and with some of our collaborative partners to look at things like bee propolis and looking at bioactive components in there. We've also done some work with our imaging partners that look at tomatoes and various other food products and seeing how they are impacted by heat stress or various diseases and characterizing where they are spatially within a plant. But really what I wanted to get to the point of is that most people that do chromatography and mass spectrometry use what we would call a 3D experiment. So that involves your mass to charge ratio and your retention time and your intensities, and it gives you this 3D plot of peaks. And that is the fundamental unit that we are looking at in a metabolomics experiment. And when we're looking at how we separate chromatographically, we might have a peak and we can get a high resolution accurate mass that can give us an indication of molecular formula. And that can match to many different things. There's lots of isomeric species out there that can be that same molecular formula. So when we're trying to determine what these species are, it can be kind of challenging because the fragmentation patterns can be pretty similar. So we're looking at this one particular little peak that we had at retention time 1.29 minutes, and it has this fragmentation pattern. Okay, let's go into a database and see how it compares to our potential structures. And those fragmentation patterns look pretty similar. So it becomes very challenging to figure out what this molecule really is. So we can look a little bit deeper by using a tool called trapped ion mobility, which can be really beneficial in separating molecules based on their size and their shape. So this is a cartoon of what we would call a TIMS cartridge, and it does exactly that. It separates molecules based on their size and their shape. So in the forward direction, we have a gas that's pushing behind the molecules, and in the opposing direction, we have an electric potential. And our favorite analogy is actually with sailboats. So if you can imagine that these sailboats are being pushed with wind behind them and they are going through an opposing water current, the bigger the ship with the bigger the sails, the further that's going to be able to penetrate through that opposing water current. So our big ions are going to be the green ones that are further into the trapped ion mobility cartridge, and the smaller ones are going to be the blue ones. And these actually could be the same or very similar masses, but if they have a different size and shape, they will be separated based on that size and shape in this cartridge. Now, this is called trapped ion mobility. They are trapped in here for a period of time. And to get them out, we can drop the electric potential in a stepwise manner. And now we have what we would call mobility separated ion packets. So this ion mobility technique is used to do this additional separation prior to going into the rest of the mass spectrometer. Now, our systems don't just have one TIMS cartridge, they actually have two in tandem. And this enables us to use a DDA method, or data dependent acquisition method, called parallel accumulation and serial fragmentation. So once we accumulate our ions in the first TIMS cartridge, we transfer them into the second TIMS cartridge, maintaining their separation. And then, as we are eluting those out of that second TIMS cartridge, in parallel, we are accumulating another set of ions. And this gives us what we have essentially 100% duty cycle and lossless ion transfer. So in a DDA type of experiment, we can do our precursor selection. And then we can synchronize those precursors with that second packet of ions. And now we can perform fragmentation that's matched with those precursors. So what we get is mobility separated and mass separated fragmentation patterns. So you can see these two red ion packets in the middle. They are of the same mass, but they have different mobility values, and thus their fragmentation patterns are going to be different. So this enables us to give an orthogonal physical chemical measurement of our analytes and enables us to help determine what these molecules are. So when we look at the mobility of that particular molecule that I showed on the previous slide, these are what we would call on the left mobilograms. So we can enhance our mobility resolution and get separation that enables us to determine what these analytes are with the additional information of retention time, accurate mass, and fragmentation patterns. So the collisional, this is what we would call a collisional cross-section measurement. So each one of these compounds is going to have a different CCS value. And this gives us additional confidence in what these compounds are. We can, in our systems, enhance the mobility resolution even further. So we do have default methods that give you separation, and you might not have full baseline separation, and sometimes you can have challenges with getting complete baseline separation, but it does give you the improvement of isomer separations in complex mixtures. So I'm going to give you some examples of what we've done using what we would call a 4D metabolomics experiment to look at licorice root. I'm not even going to pronounce it because I'm terrible with pronouncing these names. But this compound is, or the plant is licorice root. It is actually what is traditional Chinese medicine, and we have some collaborators in the Asia Pacific region. I actually did this work in conjunction with our application scientist Ryo Nakabayashi that works in our Yokohama demo lab. And what we did was use an extraction method. We actually have a partnership with a company called Pre-Omics. They actually make a lot of proteomics sample preparation gear. But this little machine on the very left is called a beatbox. And what it does is it uses sonic waves to be able to routinely and reproducibly perform extractions. So we were using this machine to extract our samples of licorice root. We can then perform a standard separation using an Agilent 1290. We actually recommend Agilent 1290s. We actually sell Agilent 1290s to go fully integrated into our TIMS TOF systems. And then depending on the type of metabolomics experiment you are using, we have a couple of different flavors of columns that we would recommend for standard workflows. So for lipidomics, we have this BioLP column, or for metabolomics, we have the BioAQ column, which is essentially what we use for this experiment here. We then perform our separations on TIMS TOF HT. We're using this with what we call a VIP HESI, that stands for Vacuum Insulated Probe Heated Electrospray Ionization Source, with the PASEF algorithm. So the PASEF algorithm involves approximately a 300 millisecond duty cycle with 1 round of MS1 data acquisition followed by 2 rounds of MSMS data acquisition. And this enables you to get about 300 hertz of MSMS data acquisition, which means that you get a lot of MSMS data, which means that you're capable of getting a lot of information for annotation capabilities. And then we use that in conjunction with the MetaboScape software, which is our software for untargeted data analysis. So this is an example of what PASEF data looks like. On the left-hand side, you can see some mobility values that are being pulled out from our chromatogram on the licorice root separation. And on the left-hand side, this is a look at all of the MSMS that we're getting across our entire chromatographic run. So every one of those red diamonds is a precursor mass that has been mobility selected and mass selected for fragmentation. So good depth of coverage in MSMS. One thing we also like to run with our systems as we're acquiring our data is a tool called RealTime QC. So if anyone's run a lot of natural product samples, and I've run a lot in my day, what we would do, or even with human samples, any kind of large cohort of samples that we're trying to analyze, we would take a pooled QC, a small aliquot of every single sample, mix it together, and inject that every 10 samples or so. And then we would overlay the TIC, the BPC, take some extracted ion chromatograms of some compound that we know is going to be present in our sample and say, is it looking good? And we'd baby that through the entire run. So we've made a tool to do all of this for you, and it will track your liquid chromatography parameters, so your intensities, your retention times, peak areas, your TIMS parameters with respect to CCS value, and it will track your MS measurements in the form of mass accuracy and isotope fidelity. And you can track it based on your pooled QC, and you can track it based on your samples. You can also take a look at your blanks in this. So this is an example of 300 repeated injections of SRM 3672 urine. So that can clog your ESI needle pretty easily if you're not too careful. And this shows that if you, at the end, we're starting to see a little bit of a drop. It might be a little light on there, but it's being highlighted in yellow. So once that starts to fall, too much, it turns red, like those couple of peaks in the middle. And you can say, there might be something wrong with my run. I'm going to stop the run and clean out my source or make sure that there's no leaks in my column or system. And then we can start up again. And it's a good way of making informed decision-making on your data so you're not wasting that precious time running a whole bunch of garbage data. So again, I will just kind of go through how we looked at these licorice root samples. We did run in both positive mode and negative mode. And just to give you a look at the number of MS features that we were detecting in a chromatographic run, each one of those little red diamonds is an MS-MS spectrum. And then we also have the number of what we call MoMA events. This stands for mobility offset mass aligned. So we, in positive mode, had 900 of these. That means we have 900 isomeric or isobaric species that give the same chromatographic peak, but more than one mobility peak, which would indicate that you have multiple species present under one chromatographic peak. There's a lot of those in this sample. And we still haven't even fully characterized this data, but we are still looking at annotating based on a number of spectral libraries that we have available. So looking at various parameters such as how tight is your mass accuracy, how good is your MS-MS score, how good is your CCS value, and all of those combined with good quality data to make an assessment of your annotation. And we did all of this by looking at a target list. So we know a little bit about natural products in the field. There's a lot of publicly available databases out there. And Ryo Nakabayashi had a really good idea of the kinds of compounds that he would expect to be present in there. So he developed what was called a target list. And we used this in the MetaboScape software to see how many annotations could we actually get from what we would suspect is present. We can also use things like spectral libraries. So we have publicly available databases like Natural Products Atlas, there's Lotus, there's Coconut, there's a number that are out there. There's the Riken database as well. And we can use these as long as they have 2D structural information in the form of an INCHI or a SMILES code that you stick into a list, and then that will enable you to predict your MS-MS fragmentation pattern and your CCS value. So we do all of this in MetaboScape, and I'll give you a little bit of a run through of what MetaboScape does. This is what does all of your feature detection, your de-isotoping, so you're not looking at your carbon-13 peaks. We can do retention time alignment. We can group our adducts and neutral losses together. So if you have sodium and potassium present or a water neutral loss, those all get combined into one feature. We do have this all set up with default processing methods, but you can optimize all of that yourself too once you get the hang of it. We also have automated methods for doing the annotation, and you can combine positive mode and negative mode feature tables together. And this will give you high confidence annotations. We use this with MetFrag is our method for doing in silico fragmentation. And then we've also developed a machine learning model, and that's our CCS Predict Pro, which can predict that CCS value for you. We have tools called correlation tables. They aren't popping up right now. They were the little boxes at the beginning that show you spectrally similar compounds. So if it isn't annotated in a library, but something else is annotated in there, you know that it's spectrally similar and it might be a very similar compound structurally. We also have, if you are looking into how these things are metabolized in a human body, we have a biotransformer module in there that can look at phase 1 and phase 2 biotransformation products. It also has a gut microbiome module in there, and the newest version even has some environmental microbes in there as well. It is a highly interactive user interface. It gives you overviews first and details on demand. So you can go and dig deep. There's a little tab up there called Compound ID where you can really do some digging into what a compound actually is. And it will connect to publicly available databases like PubChem and ChemSpider, so you can probe the publicly available space there as well. It does do statistical analysis as well, so both multivariate and univariate statistics, so you can pull up PCA plots. Like, yay, it's showing up there right now. And if you click on a spot in that PCA plot, it will actually bring you back to that feature within the feature table. Okay, so back to our target list. So Ryo actually developed a target list containing 382 metabolites that he expected to be in this licorice sample. We set our parameters to be relatively stringent. I like to see less than 2 ppm mass deviation if I'm going to match to something that's just an accurate mass. Retention time, we didn't necessarily have, so you can set that pretty broad if you don't have it. M sigma means isotope fidelity, so how close your isotope pattern matches to that chemical formula. And then the MSMS score is a cosine similarity match to what is the experimental or predicted fragmentation pattern. CCS values, we also like to see those relatively tight. 2% I would even think is a little bit wide. I like to see 1% or less if it's going to be a CCS deviation, but probably because broad natural product sample, we're a little bit wider. And then we used CCS Predict Pro on the INCHI keys or INCHI codes that were embedded in our target list, and then MetFrag as well for the fragmentation patterns because we didn't have experimental data on these compounds. The other thing we used was spectral libraries. So these were publicly available spectral libraries that contained MSMS data. The 2 libraries that we used were actually from Riken and MS-Dial as sub-list from MS-Dial. So we use those 2 spectral libraries to perform our annotations. Those did have MSMS data in it, but we still had an INCHI in there so we could predict the CCS value because there's a lot of spectral libraries out there that are publicly available, not a lot of CCS libraries out there. So our negative mode data, this is kind of an overview of the feature table for these results. This AQ score is our annotation quality scoring, so it is assessing all of these parameters up here. And I like to say green is good, so the more green on your annotation list, the more positive I am that that compound is what it says it is. It will tell you what spectral library or target list that you got it from, and it will calculate molecular formulas for you as well. So there's a lot of information in MetaboScape. It can be a little overwhelming when people first start to use it. This is where our applications team would always come in and give training. And I know that some of the people in the audience have had that training. Okay, and then, oh yeah, I did highlight that the annotations are coming from spectral library or target lists. You can see all of the individual values in there for mass accuracy and isotope fidelity and your MSMS scores. It does give you an overview of your intensities as well. So I always like to put the blanks in there. You can set up your statistical analyses as much as you like. You can measure things as pooled QCs or blanks or your actual samples. And when you do the MetFrag spectral library match, and it's not spectral library at that point, it's when you use MetFrag to predict an MSMS fragmentation pattern, and that is an example of one there, it will try and match the fragments in your spectrum to an actual fragment molecule. So it will put that on the spectrum for you. So you can make a good assessment of whether or not that's a realistic fragment for your compound. Annotation quality score. Okay, a little bit about these MoMAs and a couple examples of these, because I say MoMA events that 1 chromatographic peak, 2 mobility peaks. So we have matched to a spectral library, but those might be isomeric species in there that are giving you different positions. One is probably a different hydroxyl group on a different position on the ring, and you're getting 2 different peaks in 2 different quantities. So we can actually do quantitative analyses on these systems as well, and we can use that mobility data in there to also enhance our quantitative capabilities. Oh, I didn't leave that one up. This is that. Darn it, I was editing and I messed that one up. But you can see a little bit of these. This is my negative mode data in the background. And the little red dots, each one of those across the chromatogram represents a MoMA event within the chromatogram. And there were over 500 of these in this particular sample that we were able to pull out. And then I wanted to talk briefly about what some of our collaborators are doing. So we work with a company called Invita Biosciences. They are a drug discovery company in Boulder, Colorado, and they want to characterize all of natural product space. So they are looking at, they have greenhouses. I've been to their lab. They grow tons and tons of plants. They extract the compounds from the plants. They do initial separations, and then they perform these bioassays with particular targets to see if they're bioactive. And then they run those fractions on the TIMS TOF HT. And this is an example of some of their bioinformatics that they run. If you are a bioinformatician, our systems are very well poised for you because we like to have our data formats very open, so you can take that data and manipulate it the way that you would like, so you can do your own downstream data analysis. So each one of these little blobs here is a separate compound that has been mobility separated and mass separated. And this is mobility, retention time, and mass within this plot. So this is what you get with a TIMS TOF. You can tackle some of nature's most complex problems because we have this additional and orthogonal measurement of CCS within ion mobility. We do have quite good peak capacity and good dynamic range. Our TIMS TOF HT system has in our standard metabolomics experiments about 5 to 6 orders of magnitude of linear dynamic range, which means that you can get those smaller peaks as well as your really high abundant peaks, all at the rapid speed of 300 Hertz. One other tool I wanted to mention to you, and this is kind of going a little outside of the box because I don't know how relevant it is to natural products research or to plant research, but probably more on the life sciences, especially when we're looking at things like TCA cycle molecules. A lot of people like to derivatize their samples. So this is a new tool that we built within MetaboScape to start looking at derivatized samples in an untargeted way. So we like derivatization. It makes our compounds more stable. It can improve the separation of your metabolites, and it can improve your ionization efficiency and sensitivity. You can also cover more with one method and thus more in one polarity. So there's a lot of benefits to derivatizing your samples. The downside is that it now changes your molecular structure, your mass values are shifted, and you can possibly, if there's multiple functional groups on there, get more than one derivatization product. Now all of those compound libraries I just talked about don't work anymore. That makes your annotation really, really difficult. So what we've done is develop a tool that will take any spectral library and any reagent of your choice and enable you to predict you can test it on a structure, and it will predict all the products within a spectral library. So that would include any of the spectral libraries that I just talked about and derivatize it in silico. And then it would look for those compounds in your data set. So it enables you to have a way of looking at a derivatization experiment in an untargeted way. Last thing I wanted to touch on was a little bit of sensitivity. We do have one customer in Germany at the Max Planck Institute that is actually looking at single plant cells. And I think I actually talked to them at ASMS last year about a TIMS TOF system, and now they have one, and I'm really excited about it. So I would love to see more of this in the plant space. I'm starting with lipidomics just because this is how we've developed it. And I know that a lot of people will look at lipidomics in various types of experiments, including plants, but I see it more in the human health related fields, mostly in this particular workflow I'm going to show you is with cancer research. But the overall strategy in a lipidomics experiment is very much the same as a metabolomics experiment. So same idea, we want to have TIMS enhanced lipid separation. We want to have PASA for that clean MSMS. Our machine learning model for doing CCS prediction is a little bit different. We have a completely separate model for lipids versus small molecules. And then we also have some additional tools like Kendrick mass defect plots to be able to look at homologous series of ions. As well as that includes some automatic outlier detection in either the retention time or the CCS domain. And similar to what I was mentioning before, we can ping out to those publicly available databases to do more in-depth searching. This is just a nice example of mobility separation for lipids. So a comparison of a PCO and a PE that are co-eluting. You have isobaric compounds that are only 36 millidaltons apart. A quadrupole is never going to be able to separate these, and I don't care how high of a resolution that you have with your MS1, MS2 is always going to give you this convoluted spectrum because your quadrupole is never going to be able to separate those prior to your collision cell. So what we end up with when we use TIMS is we now have mobility resolution. So these two species are separated. The yellow one is your PE and your blue one is your PCO. And they have two distinct CCS values, which we can use in annotation. And now you have two completely distinct MS-MS fragmentation patterns. Lipids actually shows some really good examples of these. We can, in some cases, also look at structural isomers based on the position of double bonds in these chains. So they're, in one case, a little bit further up the chain, and in the second one, a little bit closer to the head group. And these ones will actually separate really nicely in mobility. I will be completely upfront. This doesn't happen in every single case. If you were to take an SN1 and swap it with the SN2 chain, you might not get as good of a separation, but it does give you some additional capabilities, even in the lipidomic space, to separate structural isomers. Now back to single cell, and I would love to see this one day with some plant research. The system that they have in Germany is different from this one, but this is work that I've been doing with the Mayo Clinic in Rochester, Minnesota, with a collaborator, Dr. Sol Ki-Bian. They have this cool instrument called a Cell-in-One, which does automated cell sorting. So it has a nice little CCD camera, and it will pick up the size of the cell, and it will measure it on the fly as it's being injected into a 384 well plate. If it doesn't meet the criteria of a cell diameter, it spits it out and it doesn't collect it. So we collected numerous amounts of cells within this 384 well plate. And then we actually set this up like a proteomics experiment. We use nano-LC. And we actually coupled this with one of our high sensitivity systems called an SCP. This is the system before the Ultra and the Ultra 2, but the system that we used in this research was the SCP. That stands for single cell proteomics. I'm renaming it the SCL for single cell lipidomics, or SCM for single cell metabolomics. And then we used MetaboScape in conjunction with this workflow to be able to do all the lipid annotations. I will say that MetaboScape also has rule-based lipid annotation in it, and I think I have a slide on what that is in the next slide. So this particular experiment, we were using human cholangiocarcinoma cell lines. I'm much better at pronouncing that one than the plant names. And then we did a simple lipid extraction with 10 microliters of methanol. That choice was more for solvent compatibility with the 384 well plate versus trying to get a full lipid extraction. Usually you need some more harsh organic solvents that would probably pull too many plasticizers into our sample, and thus we would get a lot of ion suppression there. So that means that our focus was more on the amphiphilic lipids, but as you'll see, we still got in a single cell around 160 on average lipid annotations using really stringent lipid annotation rules. So just to give you an overview of the chromatograms on an SCP, this is giving you in positive mode up to 10 to the 7 signal sensitivity on a single cell, and in negative mode, which tends to be a little less, we got about 100 times less signal sensitivity, up to 10 to the 5. And this kind of gives you an idea of some of the settings that we would use. So that 1/K0 is actually our mobility range that we set. We can modify that as well for specific experiments if we wanted to. And then this is how we would do the lipid annotation. We do have more high rate of false positive when we're just using accurate mass and isotope pattern. That gives us molecular formula information. Once we get our clean MS-MS data, we can use rule-based annotation. This is actually from the Lipid Standards Initiatives, and they're being a lot more harsh on publications recently, including one of our own from Bruker, that made us put this into our software. So now we follow some very stringent annotation rules to make sure that we're very accurate in our software to be able to do annotations. We also can now use our CCS values using CCS Predict Pro. That will give us an idea of the homologous series that are in there. Lipids are a lot easier to predict actually because the head group, number of carbons, and number of double bonds, a lot more predictable to model. And that gives us much more confidence in our species when we're annotating lipids. So I wanted to kind of go through Kendrick mass defect plots because it isn't just applicable for lipids. You can use any repeating unit you want. So if you had a series of compounds with a similar backbone, you would just have to put that backbone molecular formula in there, and then you can have a look at how those homologous series line up within that particular mass defect in the within your data. So it works really nicely with lipids. We can look at various lipid classes. We can look at it in a retention time domain. We can look at it in a CCS domain. It's a very you can manipulate it a lot to further dig into your data. So we do have a lot of comprehensive and automated workflows built in that can give you a lot of powerful analyses. So overall, with this single cell work, we were able to pull out very similar lipid profiles from 1 to 20 cells. So we knew that we were pretty consistent. And then we also ran all of this in negative mode. So negative mode, especially in lipidomics, can give us some more powerful information in the form of more informative fragments. And then finally, TIMS TOF Ultra is what we ended up moving to. We tested this again with some lung cancer cell lines and kind of leveled up and made it into a full-blown workflow. So I will have to give my great thanks to Sol Key for developing this work with us. Kind of have heard that single cell has been the next big thing to look at cell heterogeneity. Very important in cancer pathology and clinical research, but I know that it's also an important topic in the plant space as well, as if I've learned anything from my discussions with Max Planck. So we have some very powerful tools to give you comprehensive MSMS coverage from single injections. This means you don't have to repeat your injections or do some kind of iterative exclusion on subsequent samples to get the same depth of coverage that you would in one sample. We get very clean MSMS data with the trapped ion mobility, and we have really comprehensive data analysis tools so you have high confidence in what you're actually characterizing. So with that, I will leave it open to many questions, I hope. Oh, absolutely. How much accuracy in the case of the identification of metabolite by untargeted metabolomics approach? So that, I would say, goes back to what your parameters you set. The more accurate or the more narrow you set those windows for mass accuracy and isotope fidelity, your MSMS score, and your CCS value, the more stringent you make that, the more accurate your annotation is going to be. That being said, you can make that as wide or as narrow as you want. I like to see them pretty stringent, which means that you're probably going to get less annotations. You make that wider, you're going to have a lot more annotations, but they're going to be less accurate. So we always like to take the high confidence ones and not just play that numbers game of I want more and more annotations, because they might not be correct, and that can lead you down the wrong path for downstream analyses. I hope that answers your question. Okay, MetaboScape is relatively advanced compared to Compound Discover. I think we have more tools in MetaboScape versus Compound Discover. I don't usually like to comment on competitors that much, and I can't say I've used Compound Discover in too much detail. But I can say that the MetaboScape team has put a lot of effort into making the tools that you need to be the most relevant for your research. And in addition to that, I will also say that MetaboScape does have an API that you can attach to it, so you can do downstream bioinformatics directly attached to MetaboScape. So if you have your own R or Python script that you would want to run, you can do that. Is there any specific gut microbiome library that can integrate with MetaboScape? That is a great question. We do not have one available for purchase. What I would recommend for gut microbiome research is using the GNPS library. There is a lot of downloadable, publicly available information from GNPS that has secondary microbial metabolites in it. A lot of that data does come from microbiome research, and we actually do have a pretty good collaboration with Peter Dorstein's group at the moment to start putting CCS values into GNPS. So looking forward to using that to get more accuracy and stringency in those spectral libraries as well. Yes. So the feature that gives us the power is the trapped ion mobility. And if there's other systems that do have ion mobility, we do stand out in that you can very modify those mobility regions, and it's very tunable to the experiment that you want to run. So your MS, if you're just running LC-MS-MS, you're missing with this respect to if you're trying to collect MS-MS data to do an annotation that might coalute with something, then you're going to have challenges with deconvoluting those MS-MS spectra because those two compounds overlap. Then you're going to have fragments that don't match in your MS-MS cosine similarity match, and you're going to have not a good annotation. The ability to separate those compounds prior to going to collision cell means that you're going to get much cleaner MS-MS data, which means that you're going to get a better and more accurate annotation. Correct. In fact, I would say there's only a few out there that do have some kind of ion mobility.
Maybe a little less politically correct.
I'm politically correct. I'm the technical person. You can tell who's in commercial.
There's two other vendors. Agilent uses a drip tube, and then there's Waters, which uses a traveling way. They're all ion, which means they will give you a collision cross section value, just like we will give you a collision. Standard that came up first, the drip tube. And John McLean at Vanderbilt uses that. He has developed the CCS.
CCS compendium.
Thank you. And that's kind of the gold standard. We measure ours against theirs. We have to agree with John and our system to make sure our CCS values very accurate. There are other types of filtering devices. For example, I can say this, I worked at Thermo for 14 years. So at Thermo, we had FAMES. FAMES cannot give you a CCS value. It's just a major feed.
SciEx has Selexion, which is DMS, or differential mobility scanning. FAMES is field asymmetric ion mobility scanning.
It does a filter. So there's a lot of optimization that takes place. Typically, people have to make multiple injections to get the parameters set right. So it's a lot of fiddling around. But it doesn't give you a CCS value. So it doesn't offer you anything else in terms of confidence in your data, especially.
I'll comment technically on that as well, because the FAMES goes on the front end. It's after the source, so it's more prone to getting dirty, whereas any kind of ion mobility system is internal past the source. So once you get down into the ion optics, everything is much cleaner. So I would say ion mobility systems that give you a CCS are more robust. Mm-hmm. I think in order to make a good determination, these tools we give you are to speed up your analysis because your data is now so complex. You have much more depth of data that you need to go through. So it's to help you get through it faster. But if you're going to look at an MSMS fragmentation pattern and say, I'm going to do some kind of spectral matching, that you still want to have an idea of what those fragments actually mean. And we try to make our tools available to make that process easier. So like I did mention that even with the in silico fragmentation tool that we use, it will put a fragment molecular structure on top of each one of those fragments to give you an idea if that makes sense to what is actually in your sample. I still think it's very important to know what that is because I will always say you need to check your raw data. If you're going to publish, if you're going to move forward in a company with a bioactive compound, you need to make sure you know exactly what that is. Raw data is going to be very important. And to that respect, you need that chemistry background. Of course. Mm-hmm. I think Corey can speak to this one. We got some really good year-end deals.
You're in the ballpark, but so what I mean by that is we have different flavors of instruments, right? You guys have choices. So we have what's called TIMS TOF Pro 2. We have a TIMS TOF
HT.
TIMS TOF HT, and then the TIMS TOF Ultra 2. So think of those as kind of price versus performance, okay? Versus Pro 2, I would say is a very, very good small molecule metabolite lipidomics type system. The HT is geared more towards, the HT stands for high throughput. Typically, we sell those into the core lab to do more proteomics, utilizing HTs. And then if you came to us and said, Corey, we don't care anything about the instrument, we want the ultimate in sensitivity. So maybe we're done single cell or near single cell. What is your most sensitive mass spectrometer? That would be the Ultra 2. And you are right, that's for the mass spec. Then there's an LC, and there's service contract, and there's all kinds of others, right? You guys know. So, but I think at Bruker, we not with this topic, but just so people are aware, we're very flexible. Financing and to us, and parties, we collaborate a lot with people. I think that's very unique samples or access. These databases that we build, very keen on those.
And one thing I will mention about a lot of our systems is that if you bought a Pro 2, yeah, there's a bit of a hump to get into that. And that's probably our most modestly priced ion mobility system, but they are field upgradable. So if there is an improvement that's done to that system, you can actually upgrade a Pro to a Pro 2. You can upgrade a Pro 2 to an HT. And those upgrades are much less expensive than buying a whole new instrument.
Great point, Eric. You're not left behind. Our job as vendors, we are to sell instruments, come out with new technology, new ideas. You just can't get $1 million every third, fourth year. Right? The idea is to build the systems, in a modular capacity, so that when new power supplies come out, we take it over. When we as a mass spec company come up with new enhancements, maybe the craft ion ability, that's an actual example. We started with, and it turned into the Pro 2, which turned into the HT. And it was just a natural evolution of learning more about the systems and what to do, and making it slightly different in terms of the ion capacity. For example, the Pro 2 and the HT. So we're able to actually physically take that cartridge out, generation. And how we have and also resets. And it's again, I think that's very unique to us. You know, my experience when I was at Terminal was not. It's just the way it's different.
I think we have time for about one or two more questions.
Excellent presentation and good discussion also. What are the future in your company, like if you buy an instrument, a lot of times we have one, and after a certain time, like 10 years, 8 years, the warranty area, you want to get the parts, it will expire and they don't want to provide any service. So future instruments are also built like that, or is there a little bit longer lifespan? So we can
Because we have that upgradability, we'll have a much longer lifespan with respect to maintaining the parts and the components for that instrument.
What about the trends you really, other than just buying one, and then if you want to buy another software, then you really buy a new one. Is it
So
possible to add on something?
So MetaboScape is constantly updated, and it's really nice because if you pay for it, you have it. You don't have to have an annual license fee or anything like that. If you have a service contract, you get an upgrade. If you want to get an upgrade, you can purchase an upgrade. We do upgrade twice per year. The second release within the year is kind of a freebie. As long as you have a license for the first part of the year, that second one remains. You can get that first upgrade, no problem. We like to keep our software up to date, and we always take feedback for improvements as well. So that's something that we've been working very hard on to make sure that we stay with the same platform, so you don't all of a sudden have to buy another piece of software. And that's why we want to put everything in there. I will say the in silico derivatization tool is a separate module that is an additional license fee because not everybody wants to use that.
Are you expecting to add on the microbiome?
For an actual workflow yes, that is the whole goal of working with Peter's group.
Presentation and talk to you about the first microbiome. Do you see the need to combine the HRMS here more accurately here?
Oh, I would.
Yeah, as you know, microbiome cooler.
Mm-hmm.
Do you see the need to combine the here?
Oh, I would say, well, the TOF is high resolution in and of itself, but I would say the ultimate system for doing gut microbiome research, you need some kind of chromatography, ion mobility, and then quadrupole time of flight, because you want to have the high resolution and the fragmentation. Metabolomics experiments, if we're doing, yeah, for this particular system, because I'm not going to talk about the SCP, we have to dilute that like a bazillion times. But the standard workflow, if I have good references for plasma, we're looking anywhere between maybe 20 microliters. And then for your extraction, I'm trying to remember my solvent volumes. It's usually like 1 to 5 dilution there. Dry down, reconstitute, and then you're injecting like 2 microliters. Everyone likes to inject like 1 or 2 microliters. And that's why we ended up going with and recommending the Agilent 1290s. But I can give you like a standard extraction protocol if you're interested. It's probably around 100 nanoliters in total of plasma volume. And it would be, I would have to transfer that to what it would be for some kind of tissue extract. When I was working at Scripps, it was between 20 and 80 milligrams of tissue, and that's now down to like 1 to 5 milligrams for the sensitivity of the instruments. Uh-huh.
As you know, we have like two different systems. More unfortunate. And
Yes. I do actually, because I do have some really good quantitative data, and you can do PRM passive methods on this, and it does give you very good comprehensive untargeted metabolomics experiments.
PRM is now.
MRM,
MRM.
I got some good examples of that with bile acids if you were ever interested.
Join me in thanking Dr.